I'm Alena Jones, and I'm a senior in Dean Kissa's lab in the biology department. We focus on plant-based biology, and today I'll present my project, Growth and Development of Arabidopsis thaliana genotypes in gravitational stress conditions. Plants need to adapt to multiple stressors in their environments, and natural plant genotypes show variation in their responses to those stressors. Normally, shoots will grow away from the gravity vector, while roots grow toward the gravity vector. In the gravity vector, it always points down. If plants will be grown on spaceflights in zero gravity or in reduced gravity on the Moon or Mars, then they'll definitely experience gravitational stress. So it's crucial to understand how these plants respond to the stress because astronauts will need to depend on these plants for food, oxygen, and for psychological benefits. Stress-resistant plants are required in order to ensure the best growth and productivity. We use Arabidopsis thaliana because this entire genome has been sequenced of the plant, and by identifying stress-resistant and stress-susceptible genotypes, this model plant will allow us to define what genotypes are responsible for resistance to the gravitational stress. So if we can define genes responsible for adaptation to gravitational stress, then we could check or manipulate them for varieties suitable for spaceflights and space greenhouses. So the objective of this experiment was we wanted to test 100 wild-type genotypes of Arabidopsis thaliana to define the stress-resistant ones by using a 2D clinostat to simulate the gravitational stress. That confuses the plants because they do not know which way is up or down. And we hypothesized genotypes vary in their resistance to gravitational stress. So what we did, we used, as I mentioned earlier, 100 wild-type genotypes of Arabidopsis thaliana. Seeds were surface-sterilized and plated on half-strength MS agar. Plants were treated on a 2D clinostat or placed vertical for 7 days. And then we took images to measure such parameters as shoot length, main root length, total root length, number of secondary roots, number of root hairs, and number of root hairs per total root length with ImageJ software and used T-tests for statistical analyses. And almost all genotypes have been tested. 32 have been fully analyzed, and each graph here, A through D, they describe our results for each parameter. And to make these graphs, we assumed vertical controls to be 100% and estimated the percent difference from the controls for clinorotated seedlings. Here in graph A is for the shoot length, and as you can see, it was strongly affected by the clinorotation. And under the clinorotation, 31 genotypes have reduced shoot growth, and one genotype in the red, the very first one, the CIBC5, it did not have any significant difference from the vertically grown controls. Next, graph B, that's our results for number of root hair per total root length. And for this one, we observed variation. So you see positive growth, few negative, almost all of the genotypes here. And under clinorotation, 5 genotypes had increased total root length, number of root hair per total root length when compared to the controls. And this could indicate some gravitational stress resistance, which is good. Graph C shows our results for the number of secondary roots. We observed variation, again the positive to negative, among all the genotypes. We're still not sure if the number of secondary roots, if it indicates increased or reduced stress levels, so we're still working on to find that out. In graph D, this shows the results, number of root hairs. Under clinorotation, 16 genotypes did not have any difference or reduced number of root hairs, which may indicate lower stress levels. And based on our data from these 32 genotypes, just one genotype, CIBC5, the bar in the red, it has potential for gravitational stress resistance based on shoot, root, number of secondary roots, and number of root hairs per total root length data. Thus, the genotype CIBC5 is a really good candidate for future space experiments with plants. Our further studies will aim to identify genes involved in developing resistance to gravitational stress, and in the long term, these studies will help us to select or manipulate plants for spaceflight missions. Thank you so much for listening.